Okay, so welcome and thank you all for coming and joining us today. Don't know if it's the afternoon or the morning, but welcome either way. So again, my name's Helen. I'm part of the outreach training team that's delivering this online training series. So just a recap of the course objectives of the whole series. So this is a 7-module series where we want to tell you what Ensembl is, the different types of data you can get in Ensembl. We're going to show you how to navigate the Ensembl browser website and importantly, where you can go for help and documentation. So if you've attended any of our previous sessions, then hopefully you've learned some of that so far. And then again, this is the third of this webinar series. This is me in red. So previously we've had Emily delivering the introduction and Denise delivering telling you about Ensembl genes and transcripts. So if you've got any questions throughout this webinar today, there's a chat box in the panel at the bottom, and you can expand that and type your questions. I've got Emily and Denise in the room with me today, so they'll answer your questions live as I continue to talk. There's no threading with the questions, so if you start your questions and answers with your own username first, then we can help. Then we can do some threading and you can figure out who's answering whose question. That would be useful. Okay, so the structure today, as most of you have been to the previous sessions, you'll know that I'm going to give a presentation, and today I'm going to explain what BioMart is and show you the steps to making a query. Then I'll do a demo, a walkthrough using the tool, and then the exercises will be provided to you after the course. They'll be on the Train Online course page, and the solutions will be there as well. So here's that Train Online page. There's a section for data export with BioMart, which is today's session, and the links to the exercises and solutions will be provided in the panel on the left. I'll upload a video of today's webinar, and that will be provided in the middle. And then on the bottom right, the next page will be to the exercises from the BioMart page. And again, if you need any help with the exercises, there's quite a lot of exercises coming with this session. The exercise solutions will be provided. Also, we have the Facebook group where you can discuss with everybody. I hope you'll use that. Otherwise, you're very welcome to email the help desk and we can help you with any problems that you're having on the course. Okay, so let's get started with today's course, which is Data Export with BioMart. The outline of today's course, firstly I'm going to tell you what BioMart is, and then I'm going to walk you through the process of using BioMart. It's a 4-step process, and that will become clearer as we move on. Then I'm going to do a demo with an example BioMart query and show you how to use those steps, and then at the end I'm going to round up with where to go for help and documentation. So first of all, what is BioMart? It's a tool in your browser that lets you export data from the Ensembl databases without needing any programming to access that information. The way that you build your queries is just with a few mouse clicks, so it's quite simple. And then the output of any BioMart query is accessing the data, and then will give you a table or FASTA sequences which you can view online or download. So why might you use BioMart? Kind of seems like a strange, just a strange word to start with, so what am I trying to tell you? So BioMart is a tool that lets you access data that you might have seen in the Ensembl browser, but if you want to access lots of data at once, BioMart's really useful. So if you're looking at lots of genes and you want to do ID conversions, you can do them all in one go with BioMart. Or you can look up gene locations of lots of genes at once, or download sequences of lots of genes at once. Or if you want to export large amounts of data in one go, you can look at all of the variants from one gene and download that in one table. So we have BioMart available in Ensembl, so that's where I'll show you in the demo today. We also have availability of BioMart in Ensembl plants, in Ensembl fungi, Ensembl metazoa and protists. So I looked at the results of the poll last week, and it looks like most of you are working with vertebrates, so you'll be using Ensembl, but a few of you were looking at fungi and metazoa as well. Unfortunately, for those of you working with bacteria, the databases are just getting too big that BioMart isn't really able to cope with all of that. So sorry, it's not available for you. So now I'm going to introduce you to how you build your BioMart query. It's a really simple 4-step flow. So first of all, you choose the data, and that's the database and species. Then you pick your filters, so that's limiting the data set. So narrowing it down from just the species. Then you choose your attributes and then your results. So I'm just going to talk to you a little bit more about what each of those is. So you choose your database and your data set. So the database is the specific database that we have in Ensembl for genes. We have different databases for genes, variation, and regulation. So you'll choose the database depending on which or what the main focus of your search is going to be. So if you're looking at genes, you'll go to the genes database. If you're looking up variants, you'll go to the variation database. And then the next step is to choose the species of interest that you want to look at. So after you've chosen your species, you want to narrow your search by choosing combinations of parameters, and these can be, so for example, if you're looking for genes within a certain region, you can pick region, you can pick biotypes, list of IDs, the gene function, different phenotypes. You can pick more than one filter, and as you sort of choose different combinations of filters, it's where they overlap. So it's an AND function. So if you pick a list of protein coding genes within a certain region, you will only get protein coding genes of that region. And then the next step is to pick the attributes. So this is the data that you want to get out of our databases. So this is sort of the results table that you're trying to build here. So if you want to find IDs, features, sequences, orthologs, paralogs, these are all things that we can give you in BioMart. So basically, you can access any of the data that we have in our databases through BioMart, and you pick the attributes based on the data that you're looking for. And then you click results, and you can choose different outputs depending on what you want. So if you're getting a table of results, you can see that in your browser in HTML. And you can also download different table formats as well. And then we have FASTA for sequence, which you can either view online or download. So a lot of people find it quite confusing to tell the difference between filters and attributes. So I'm just trying to break it down a little bit more. So filters are your search terms. So that's the information that you have that you want to find out more about. And then the attributes are the answers that you want to get. So I've got two little examples here, and when I give a workshop, normally I ask you what you think the filters and attributes might be. So I'm just going to go through for the first one. So for these two human Ensembl genes, what are the HGNC symbols? So of this query that we have, which do you think the filters might be, and which do you think the attributes might be? So I'll just give you a second to think about that. Okay, so the filters were the genes, so that's the information that we had, so they were our search terms, and the attributes there was what we wanted to find out. So we wanted to find out the HGNC symbols of those genes. And I've got another example here. So list the variants, the RSIDs of the CFTR gene. So what's the filter there and what's the attribute there? I'll just give you another few seconds. Okay, so hopefully you got this right. So the variants here are the attributes. So that's the information that you wanted to find out. And the CFTR gene was what you would use as your filter. All right, so just before we start our walkthrough, this is where you find BioMart. So it's up here in the top panel of the Ensembl homepage. If you're working with the older human assembly, you'll need to go to the DRCH37 page at this URL and use BioMart from there. And the same is true for the different taxa pages. If you want to use a plants ensemble, you'll go to plants.ensemble.org and use BioMart from there. Okay, so we're going to start the demo part of this. So the query here is that we want to find out the entree gene IDs and the function via Go terms of these 6 human genes I have here. And we also want to find their cDNA sequences. So just going through the 4-step flow here, the database that we're going to pick is genes. So we had a genes database, a regulation database, and a variation database. But because we're looking at genes, we're going to pick the genes database today. And because these were human genes, we're going to pick a human species dataset. The filters that we have, so the information that we have in our query are genes. So we're going to filter by the genes that we have. And then the attributes was the information we wanted to get out. So this will be the table that we create. And then the results is that we're just going to view the table in the browser, the HTML table. To get sequences, we'll need to go back and change our attributes, and then we're going to download the sequence to a file. So let's get started. So I'm going to start here. This is the Ensembl homepage, and as I mentioned before, you can find BioMart in the panel, this header here at the top. And pretty much every single page in Ensembl has this header, so you can always access BioMart no matter where you find yourself. So just click on that. And we can see that there's a button here for picking the database. So as I mentioned, we were going to choose Ensembl Genes. And you can see it says Ensembl Genes 84 there. And that will change as we release new updates, which Emily talked to you about. So every release will update BioMart. So in a few months' time, when we make release 85, BioMart will be updated to Ensembl 85. But if you want to use Ensembl, if you want to use BioMart from a previous release, you can go to the archive site and do that. Okay, so we've selected Ensembl Genes, and now we're going to choose the data set, which I said would be human. You can see there are 5 at the top here, and these are just the most popular. Human is in there. If you want to pick any of the other species, you can just scroll down and find the species there. I'm going to pick human. And you can see on the left side here that we've got some more buttons. So filters has appeared, and so has attributes. So step number 2 is to pick our filters. You can see there are 7 different types of filters here. So for example, region, gene, phenotype, and the other ones that you can see. Because we had a list of genes, I'm going to expand gene there. But had you wanted to look at a specific region, you could expand that. You can pick a chromosome or specific base region. But let's look at gene. Okay, so I had a list of genes. I'm going to paste them in this input external references ID list box. And there are the 6 genes I've just pasted in. You could also have them comma separated. But these are not Ensembl gene IDs, which this drop-down box shows. So you need to make sure that this drop-down box has the same ID type as the one you've pasted. So I'm going to click on associated gene name. Okay, so I have 6 different genes in here. I'm going to click count now. And as you can see, next to data set, it's given me a number. And this number is 6 out of 66,000 genes. So this just confirms to me that I've added these filters now, and that's narrowed my data set. 6 genes out of all of the human genes. Okay, so now I know that I'm just searching those 6. I can move on and click my attributes. So I'll just click this button here. And as you can see, underneath attributes, it already has Ensembl Gene ID and Ensembl Transcript ID selected. So any table that you create subsequently will always have those unless you unselect those. Okay, so now looking at the attributes, you can see there are 6 different types of attributes here. There's features, structures, homologs, 2 different variant types, and sequences. You can see in features we have gene, external, proteins domain, and families. So I'm going to expand gene first of all, and this is where you can see the Ensembl gene ID and Ensembl transcript ID pre-selected. We don't want those in our search, so I'm going to untick those. But we did want the associated gene name, because that was our input. So if you want to have a column that was the same as your filter, then you should always click the same type as your filter. So we had associated gene names, so I've selected that again. We were also interested in the Entree gene IDs. So Entree gene IDs are external to Ensembl. They're a synonym that we bring in. So they're under external references, and I find it there. And I also wanted the Go terms, which are also external, and I'll click on those 3. So those are all of the attributes, so those are all the columns in the table that we wanted. So now I'm ready to click results up here at the top. And that will just take a second to load. So it's just loaded a preview table here. But you can see for each of, well, you can see for this this gene that I inputted, it has an Entree gene ID, and then it has multiple Go term accessions. So for this single gene, there are multiple Go terms, and that's why there's lots of rows. There are only 10 rows displayed on this page, though. This is just a preview table. If you want to view all rows, you can use this drop-down box. But before we do that, I just want to highlight this unique results only button. So this button just makes sure that there are no duplicate rows in the table. Okay, so just click all, and doing that will open up a new browser tab or a new window, and the entire table of results will load in that. So this is the entire table here of the 6 genes. They're Entree gene IDs, and then all of the different Go terms associated with each of those genes. Okay, so let's go back to our search query. As I was saying, for sequences, you can keep your data set and filters the same. We'll just click on attributes, scroll to the top, and then we're going to click the sequences button here. I'm going to expand sequences, and then we wanted the cDNA sequences. And if you look at this image here, you can see as we move from the default of protein towards cDNA, the red boxes change. So this is just which part of the sequence you're going to be, or which part of the gene you're going to be selecting in sequences. And then I'm going to scroll down to header information, because we'd also like to show the associated genome here, and not the Ensembl gene ID. Okay, so I've selected associated gene name and cDNA sequences. I'm going to click results again. And as you can see, we've got the FASTA sequence header at the top, and then the FASTA sequence below. But again, this is just a preview. So if you wanted to view all of that in your browser, you could just click all. But I'm also going to show you here, you can download the file as a compressed file, a compressed web file that will notify you by email, which I strongly recommend if you have a large file. So if you're downloading a lot of data from Ensembl, I'd recommend that you use the notify by email. But today we're just going to download a file. Sequences only come in FASTA format. We'll click unique results only and then go. And then when that file's ready, it'll appear in my downloads folder, so you can see that there as BioMart export. So that will be, just open that. So that's created a text file there of all of the sequences of those 6 genes. Okay, so that's the end of my demo. I'm going to go back to my presentation. Hopefully Emily and Denise have been answering all of your questions as I went along. So that's the end of the BioMart walkthrough. I'm giving you lots of questions to do before the next course. I think on some BioMart's sort of the best way to learn about it is to do it. There are 6 different questions, and I'm going to sort of recommend that you do all of them, but some of them might suit your work more than others. So obviously make sure, if you're not going to do all of them, you can look through the 6 and see if some of them might suit your needs best, and you could try those first. Next week you've got Denise back again. She's going to talk to you about variation data in Ensembl and tell you about another tool that we have, which is the VEP. So that's next week at 4 o'clock, the same time. And then just to reiterate where you're going to find the exercises. The exercises should already be loaded now. No, they'll be loaded shortly, sorry. And yeah, they'll just be on the Train Online course, and there'll be a drop-down box just here, like the plus, there'll be a plus there. And then tomorrow I'll upload the video here, and you can find the exercises there on the next page as well. Again, if you need any help with the exercises, the solutions will be provided when the questions go up. You can also discuss in the Facebook group, and please do email us if you need any more help. And then just generally, this is the help and documentation access. So we have lots of other tutorials on Ensembl. We also have a YouTube space where we have lots of different videos. There's also another BioMart video that I did a few weeks ago, so that's a fairly recent one. Please do email us if you have any questions about Ensembl. We have a couple of public mailing lists as well. And please follow us on Facebook, Twitter, and we have a blog. So this is, we use the blog to talk about releases. We use Facebook to talk about, highlight different features. So if you're interested, please do follow us. These are the different publications. So if you want to find out more about Ensembl, you can read our papers. And if you do use Ensembl, please cite us. This is the Ensembl team last summer. We're still growing, so that's not even got me on it. And then I'd just like to acknowledge everybody on the team and all of our funders. So just to wrap up, if there are any more questions, I'll open up the chat box now and see if I can answer them live. Okay, so I'll start with Roberta. So in which section of BioMart annotation can I find Entree ID? So if you mean in filters, if you have Entree IDs as your filter, you can find that in input external references, as I just showed you. So there we've got Entree gene IDs and transcript names there. And that. Okay, so that's if you have gene names, then you go into filters, you expand gene. So as there were the 7 different options there, you expand gene, and then external references ID list is where you put all of your gene IDs. For attributes, again, they're in features and then gene, and Entree gene IDs are external, so there it is, it's still checked there. Right. Okay, I'm going to wrap up now. Thank you everybody for coming today. Hopefully we'll see you at Denise's webinar next week, learning about variation in Ensembl and the VEP. Thank you very much.